So, hello, welcome everybody. My name is Dr. Mihic. I work here at Real Madrid. I'm the medical director. One of the topics of great interest to all of us here is Elite Athlete Gut Microbiota, which is different from non-elite athlete microbiota. It's really interesting. It's a topic that's growing. I started testing some of my patients, and I'm still looking for interpretation. So we have the great privilege of welcoming Dr. Paul Cotter, who's coming to see us from Cork, world authority on this topic, and he'll tell us stuff that we all need to know to apply. Se lo digo en español también. Soy Dr. Mihic, médico director de Real Madrid. Tenemos el privilegio de aceptar aquí, a invitar aquí a Dr. Paul Cotter, que es un experto mundial de microbiota de atleta de élite, como lo que nosotros tratamos. Ricardo, ¿tú tienes algo que comentar?
Gracias, Nico, and good afternoon, everyone. My name is Ricardo Rueda. I'm the global head of the Mobility and Metabolism Science Platform at Abbott Nutrition Research and Development. And in collaboration with Real Madrid, our Abbott Nutrition Health Institute is endeavored to promote health science worldwide through providing educational events such as the one that we have today. And I have the pleasure also to introduce Dr. Paul Cotter, that, as Nico mentioned, is with us today. Dr. Cotter is head of food bioscience at TIGAS, principal investigator at the large Irish research centers, APC Microbiome Ireland, Vista Milk, and Food for Health Ireland. He coordinates the European Union project Microbiome Applications for Sustainable Food System through Technologies and Enterprise, MASTER, and is co-founder of SecBiome. He's a molecular microbiologist with a particular focus on the microbiology and microgenome of foods, food chains, and humans. His group has pioneered the study of the role that the microbiota plays in athletes' health and performance. Professor Cotter is author to over 350 peer-reviewed articles, is included in the Clarivate list of most highly cited researchers for 2018 to 2021, and is the field chief editor of Frontiers in Microbiology, the largest and most cited journal in the field of microbiology. With that, I pass the word to you, Dr. Mihic, to introduce the first question.
I think it's time we speak to Dr. Cotter. He came all the way from Ireland to see us, so welcome again. And you know, the burning question for us here is obviously what are the characteristics of the elite athlete gut microbiota that's different from, for example, mine?
A good question. So let's address that. Thank you. So I'm going to cover that over the next couple of minutes, tell you about the microbiome of elite athletes. But before we get into that, it's important to first introduce what the microbiome is and why we should study it. Okay, so what are the characteristics of an athlete gut microbiota? So as I said, I want to introduce this topic to you because I'm aware that while a lot of people are becoming interested in the microbiome and microbiota in recent years, they're not always fully aware of why they should know more about it. So the microbiome, its numbers are really very impressive. So if you think about the human body, there are 100 trillion different microorganisms on and in our body. So 95% of them are in our gut, the vast majority of them in the large colon, or in the large intestine and the colon. And when you think about the genes that are present within all of those microbes, they are all different. And that contrasts with our human bodies because every cell in our body has the same genetic material. So in fact, we have 150 times more genes encoded by bacteria and other microorganisms in our gut than we have human genes. And there's all sorts of other impressive numbers relating to the fact that there are over 10,000 different species in our gut. The weight of those microbes corresponds to about 2 kgs in weight. Critically, in contrast to our human genome, which isn't so easily manipulated and we wouldn't really want to, we can quite easily change the microbiome in certain circumstances, and I'll be returning to that later on. Equally importantly, almost 90% of diseases can be linked in some way back to the gut and the health of the microbiome. And there's all sorts of other impressive numbers that you can read for yourself there. Now, what's the difference between the microbiota and the microbiome? Well, often you can use the two interchangeably, that there isn't so much of a difference. But if you want to get into the fine detail, the microbiota refers to all of the living microorganisms found in a particular environment. So that would include bacteria, fungi, small eukaryotic cells and protists, phage and other types of viruses. And the microbiome differs in that it relates to all of those individual microorganisms, but also the genetic material that's encoded therein, the proteins that they produce and other metabolites, and indeed the environmental conditions itself, so the pH, the salt levels, and so on and so forth. But in general purposes, when you're talking to a person on the street, they won't know the difference between them, so let's not get too bothered by the differentiation today. What is important is that there has been a revolution in this field over the last 20 years. And that is because in the past, we as microbiologists very much relied on old agar plate approaches that were used by Louis Pasteur over 100 years ago, and broths, and the reliance on trying to grow those microorganisms in the lab in order to study them. But as time went on, it became more and more apparent that there are lots of microbes in different environments that we can't grow very well, and that relates to soils or the microbes in fermented foods, but is particularly pertinent to the human gut, because that's an anaerobic environment, so a lot of the microbes that are in our gut, as soon as you take them out of that environment, they die. They sometimes require a certain pH, they like bile, they might like some other conditions, they may not like to grow in isolation. You may need to have consortia or groups of microbes to help one another grow, and that really doesn't happen on a Petri dish when you spread them all out. So there is no one agar plate that will grow every microorganism under the sun, and ultimately only a small percentage of microbes are easily cultured in the laboratory. So how do we get beyond the tip of the iceberg to see the entire picture? Well, we can do that increasingly by using DNA sequencing. And that relies on the fact that all microbes have genetic material. We don't need to grow those microorganisms in order to extract that genetic material. So we can take a fecal sample, or a skin sample, or an oral sample, or any other environment we want to study, and without trying to grow the microorganisms, take the DNA out of those bacteria and study it. And by doing that, we can tell what microorganisms are present, but indeed go much further to predict what those microorganisms are doing in our gut. And I'll explain a little bit more about that as we go along. Another term I want to introduce is metagenomic DNA, and that relates to when we take the genomic material from all of the different microorganisms in combination, so it's not just the genomic material from one bacterium, it's the metagenomic material, all of the DNA from all of the microbes in that particular environment. So, without wanting to get too much into the technicalities, I think it's important to have an appreciation of how we study these microbial communities and how has DNA sequencing really revolutionized our understanding. So first of all, it's critically important that we can efficiently extract the DNA from that sample and do it in such a way that it's representative of the entire community. But let's assume that happens and we have the right kits and you're dealing with an organization that knows how to do this properly. After that, there are two main flavors of analysis. There are others, but if you know the two of these, then you know most of what you need to know. So the first type of sequencing, which was, I guess, probably cutting edge 5 years ago, it's still used a lot, but is kind of increasingly regarded as the more cheap and cheerful analysis. That's referred to as amplicon sequencing, or 16S sequencing in the case of bacteria. So you don't need to know what 16S is, it just refers to a gene that's present in all bacteria, not just those in the gut, but that in every environment. And that gene is very useful for a number of reasons. So you all know about PCR. Increasingly in recent times, all of the world knows about PCR since COVID and COVID testing. In this case, PCR is very important because we take advantage of the 16S gene in bacteria, has very conserved regions and very variable regions. We take advantage of the conserved regions because we can have PCR primers that bind to these and initiate the PCR reaction, so this will work for every bacterium. But what's nearly more important is the region in between is hypervariable. And in some ways, that acts as a barcode or a QR code in order to let us know the identity of the bacterium that we're sequencing. So if you multiply that up, instead of just sequencing one 16S gene, but you do it for thousands and millions of microorganisms from an environment, and then put that sequence data into a database to figure out what that DNA sequence means, we can begin to identify what microorganisms are present down to the genus level. I say genus level, and I emphasize that because we can do better. But genus level tells us how many Clostridium are in my gut, how many Lactobacillus are in my gut, and that's important because you might want more of one and less of the other, and you can start to compare different individuals or see which microorganisms are more dominant in healthy versus unhealthy, and so on and so forth. However, increasingly now, what's the cutting-edge approach, and the one that we use routinely in our lab, is to carry out what's referred to as shotgun sequencing. So shotgun sequencing takes the same starting material but treats it differently, in that instead of just being interested in one gene, we're interested in all of the genes from all of the microorganisms. So hence the term shotgun, because we sequence them in a random fashion. We don't care if it's just a 16S gene. And by virtue of taking that approach, we start to get information about all of the different genes and the pathways that are encoded therein. So how many genes associated with toxin production? How many genes associated with short-chain fatty acid production? These are useful metabolites. How many genes associated with all sorts of different biosynthetic pathways, and so on and so forth. And you can, again, begin to compare healthy versus unhealthy people, or infants that are born in different ways, elite athletes with good diets, elite athletes with not so good diets, and so on and so forth. Importantly, as well as getting the information about the genes and pathways, coming back to our taxonomy, or the names of the microorganisms, we can go further. So not just to genus level, but actually to species level. And therefore, we can tell whether we have a Clostridium botulinum or a sporogenes in our gut, which is important because one can kill you and one is harmless, or whether we have a Lactobacillus plantarum or a Lactobacillus salivarius, both of which are good, but some are better for one scenario versus another. This data can be used to do all sorts of other things as well that I won't get into, or maybe can talk about later, but it really is a more powerful approach. So why can we do more shotgun sequencing now? And why has the field continued to evolve beyond when we just first began to do this sort of analysis? Well, it really comes down to putting together a jigsaw. So if you imagine your microbiome as being represented by this image, so you're taking a snapshot of your microbiome, if you can get as good a representation of that picture as possible, you'll have a better understanding of your microbiome. So if you think of a piece of a jigsaw that has thousands of pieces, the more individual small pieces you have, the better chance you can put most of the jigsaw together and begin to see what's happening. So a lot of the DNA sequencing instruments facilitate this. So we purchased an instrument back in 2009. This was our first high-throughput DNA sequencing devices. We thought it was revolutionary, and we thought it would last us forever, and we would never need to buy another DNA sequencer again. We threw it on the scrap heap 3 years later because the technology has evolved so quickly. And I'll show you why. In fact, some of these are starting to appear in museums now. So this will generate 1 million bits of jigsaw, which is impressive. Yeah, it's a large number. But the next instrument we bought generated 4 million bits of jigsaw, and then 20 million, and then 30 million, and then the next one, 1.2 billion, and the latest instrument we incorporated a couple of months ago, 20 billion DNA sequence reads. So as we're going along, the sort of detail we can now get into this picture now relative to what we could get with this one, there's no comparison. So I've been talking about small bits of jigsaw. The alternative strategy is where you take bigger chunks of jigsaw. So you all know of the sort of jigsaw that very young children put together. So there might be only 4 pieces, and you put them on the floor, and they're very large. So there are DNA sequencers that are similar. They generate much larger bits of jigsaw, so longer stretches of DNA, but much fewer of them. So 4 million or 20 million, for example, for these 2 instruments from Oxford Nanopore. So these on their own won't be enough to give you a good picture of your microbiome, But in some instances, putting together lots of small pieces and lots of large pieces can improve your picture as well. The other reason why people like to hear about these DNA sequencers is that they're portable, and this one in particular. So this device is about so large, and so you can bring it out into the Amazon rainforest or into a field or into a stadium, and as long as you can get DNA of your sample, you can put it in here, you can connect it up to your laptop and do real-time DNA sequencing. So the future is arriving, and these sort of instruments are increasingly being looked at in terms of doing microbiome testing of food production facilities. We led the National Coronavirus Sequencing Initiative, and we used these sorts of instruments because we didn't want the centralized sequencing. We wanted all of the hospital labs to have these sequencers, and then they sent the data to us to help to interpret it. So some other terminology that I want to introduce you to, because it's used very frequently in microbiome research, and that relates to diversity. And these are terms that we unashamedly robbed from ecologists. And so for very many decades, ecologists have been talking about diversity, and for example, alpha diversity, where they count how many species are present in an environment. So again, you have this rainforest, you have lots of trees, lots of animals, lots of insects, frogs, and all sorts. And you compare that with a low diversity environment, such as a Sahara Desert, or you might imagine an agricultural field that has one crop in there. In addition to counting how many species you have in an environment, which is represented by alpha diversity, you can also look at beta diversity. So rather than looking at both of them in isolation, you can now begin to compare the two different environments. And again, it's very clear those two environments are very different. Now, we introduced those terms on purpose because it's increasingly apparent that when it comes to the gut microbiome, high diversity is good. And that makes a lot of sense, really, if you think about it, because if you have lots of different microorganisms in the gut, if something happens to disrupt them because you take an antibiotic, or you're under stress, or you're a marathon runner and you've overdone it and you have inflammation in your gut, then at least the microbes that are present, if one is killed or something happens to it, there are lots of other microbes that can do a similar job, and the environment doesn't collapse or fall apart. Low diversity microbiomes are associated with lots of different diseases increasingly. So things like colon cancer, obesity and overweight, inflammatory bowel disease, and several others. So having a good microbial diversity is to be desired. And diversity doesn't just relate to the species or the names of the microorganisms, but also you want high diversity with respect to the genes that are encoded within it and the different functional potential, as well as the different metabolites that are produced by the microorganisms therein. So we've talked about the names of the microorganisms, we've talked about diversity, and we've kind of started to touch about the pathways and what the microorganisms can do. So to expand on that here now, and what are the links, some of the links, between the gut microbiota and health, and this is just a short snapshot. So microbes within our gut can contribute to the production of vitamins in situ in your gut, so you don't need to consume those vitamins. They help with digestion and absorption of different nutrients. They stimulate your immune system, so infants that are born through C-section typically have a less developed gut microbiome and are more susceptible to suffering from autoimmune diseases later in life, allergy, asthma, psoriasis, and so on and so forth. They can control disease-causing microbes, because if those microbes are coming from an external source, from a food that has been contaminated, if you already have a high diversity and very resilient microbes, they can compete with those incoming microbes for nutrients and prevent them from colonizing your gut. They can contribute to the efficacy of energy harvest, help the cells in your gut, the epithelial lining, to develop and proliferate and differentiate appropriately. And they can even send signals to our brain, so the important gut-brain axis. So you have compounds that can be produced and absorbed within the epithelium or trigger the vagus nerve. And a lot of studies have been shown by my colleagues back in Cork relating to how having the right microbes can make you more resilient and less susceptible to stress and other disorders. So this is dictated by what microbes you have, what they do, and your microbial diversity. So now I'm going to touch on modulation. So modulation in terms of what we do in a lab to determine how we can change the microbiome, but also to take that from the lab and actually apply it in the real world setting where we try to change the microbiome for the better, or when somebody is exposed to a disruptive agent that disrupts their microbiome, how can we begin to modulate it again to make it healthy again? So when we carry out such studies, in the laboratory, we can use in vitro or desk type studies or ex vivo studies. These are studies where we take fecal samples from donors and we study them. So we put them into fermentation units and we add different compounds to them and see how the microbes in them react. So it's a quick way of checking the response of the microbiome to different factors. We do different animal studies, and around the globe, different animal models are used, including rodents and pigs. Pigs are a good model because the gastrointestinal tract is very similar to that of a human. Something that you might find interesting relates to the use of germ-free animals. So if an animal is born through C-section and is reared in such a way that essentially it's in a bubble and all of the microorganisms from the environment are kept away from it, it essentially will have no microbes in its gut. And that's useful for experimental approaches because you can add in a microbiome and see how it impacts on health in a very controlled way. Or you can even take a fecal sample from a lean person and from an obese person and put it into the animals. And it's been shown that when you do that, the microbiota, the fecal material from an obese person, can make those germ-free animals obese, thereby proving that the microbes in your gut are responsible for that effect. And then human studies. And the human studies can be in different forms. So they can be cross-sectional studies where you take a group of society, elderly people versus younger people, different groups of athletes in different sports, and compare and contrast their different microbiome profiles. Or you can do a longitudinal study where you take individuals and you study them over time, either through an intervention, so you give them a particular food, or you ask them to engage in a particular exercise regime and see what happens to their microbiome over time. And these have the ability to impact on the microbiome or allow us to study how these different factors impact on our microbiome. So some of the factors that we study are antibiotics, which in general have a detrimental impact, especially broad-spectrum antibiotics. And this is really important because we've talked to medics from different team sports previously that on first sight of seeing an athlete with a small cut, for fear that that athlete will develop an infection and not be available for a match, they immediately prescribe antibiotics. And that really makes us very scared and very nervous because we're aware of the detrimental impacts that those are having on the gut microbiome that will have much longer lasting effects than the small risk of infection. Other things that can have a detrimental effect are stress or the environment. Birth mode can be positive or negative, as I mentioned earlier, depending on what the birth mode is. Breastfeeding is regarded as being very much desirable. And there are other interventions. So you can have somebody take a probiotic, and a probiotic is a live microorganism. You're familiar with probiotic yogurts and supplements that are taken with a view to enhancing health. What I will emphasize here, though, is probiotics are different. So if somebody tells you take a probiotic to improve your health, the first thing you should ask is why and which probiotic, because not all probiotics are made equally, and some have very good science behind them, others have very poor science. Others, there's lots of evidence relating to helping an individual to prevent traveler's diarrhea, and others relating to IBS. So you want to make sure you take the right probiotic for the right circumstance. Prebiotics are nutrients, carbohydrates typically that are used by gut microbes. They're not used by our body, but they're used only by the good guys in our gut. So they're selective to enrich for the desirable microorganisms that are in the gut. Symbiotics are kind of a combination of both, so where you take live microorganisms and a carbohydrate source or an energy source and consume them together. Postbiotics are relatively new to the market, so you might not be so familiar with those. So a postbiotic is very often a probiotic that has been heat inactivated. The merit is that you no longer need to keep it refrigerated because you no longer need to keep it alive. So you'll have dead microorganisms in there, or you'll have the metabolites from the microorganisms from before they were heat inactivated. And sometimes those alone, you don't always need to have the live microbes in there in the food in order to bring about the beneficial impact on the gut. Diet can bring about very beneficial effects. Very many people in my lab are very interested in fermented foods and how different fermented foods can impact health, and fiber is another key one. And I underline exercise because if somebody were to try to give a presentation like this 10 years ago on this topic, you wouldn't really have been able to find anybody to provide any evidence about it because nothing was known. So all of this research that I'm going to be talking about subsequently has been developed during that period of time. But I'm going to give it away and tell you, yes, exercise is important. And this is our first example. So in fact, it was 10 years ago. So this was our initial elite athlete study. It wasn't with a soccer team, it was with a rugby team. It was with the Irish rugby team. So in Ireland, rugby is quite a popular sport. It's one of the few sports that Ireland are actually very good at. And so it was quite a coup in order to get access to these players. In fact, the students were very enthusiastic to engage with them. These are sex symbols and superstars. And so we didn't need to work hard to get volunteers to get involved in meeting these people until they had to deal with their fecal samples, but that's a different matter. So what we did here is we were interested in studying them in order to establish for the first time what is the relationship between diet, exercise, and the gut microbiota. And we collected lots of other data as we went along. So because the athletes were preparing for the World Cup, there were a larger group together before the panel was shortened. So 40 of them were training together, all male between 18 and 37. Now at that time, there was a lot of debate regarding how BMI might influence the microbiota and what was the relationship between the two of those. And because, again, I'm aware not everybody here might be so familiar with rugby, but in rugby, you have the forwards who are really big, strong guys, but not very fast, typically. And you have the backs who are the leaner guys who are the speedsters. So because they had quite different BMIs, we thought we had better get some controls from the general population that also had high or low BMI as well. And then we collected lots of data relating to from blood samples and food frequency questionnaires and fecal samples and so on and so forth. And the key take-home message, the first thing we saw was that alpha diversity, and this is one metric, one way in which alpha diversity can be analyzed, was much higher in the elite athlete group relative to either of the two other groups. So straight away, this showed us that we're onto something and there's something really interesting happening here, and made us start to think what's going on and why might this be significant. We also looked at beta diversity. To remind you, beta diversity is when we compare different environments. So each dot here represents all of the thousands of little bits of jigsaw and all of the data from one individual all condensed down into one single data point. And so if you have 2 points that are far away, the microbiome is quite different. Or if you have 2 points that are quite near one another, the microbiota is quite similar. And immediately, you can see that all of the controls, the blue and green, are over here, and all of the athletes, albeit more dispersed, are further away on the right side of the graph. I will draw your attention to one exceptional black point. So one of the athletes had a microbiome that was much more similar to that of the general public. And we went back and looked at that individual's food frequency questionnaire and saw that while they were elite athletes, they weren't elite in terms of their diet. And ultimately, they got the diet that they deserved. Now, fortunately, as a consequence of this athlete was converted and rapidly began to change their diet, and in fact is still playing now at quite a senior age, considering he's a rugby player. So beta diversity shows differences between the elite athlete microbiota and controls, and there are exceptions, as I pointed out. So a little bit more detail about the individual athletes and the individual microbes that are associated with those elite athletes. So some notable examples are Akkermansia. So again, if you asked a microbiologist what's an Akkermansia 20 years ago, not very many microbiologists would know, never mind people in the general public. And its importance has been realized through DNA sequencing and is very frequently associated with a lean phenotype. This is a microorganism that's found in the lining of the gut, in the mucus layer. And in fact, some companies are now trying to develop this as a biotherapeutic compound going forward. Eubacterium and Faecalibacterium are other unusual microorganisms. And these are important because they produce short-chain fatty acids. I'll tell you later why that's important, but those are key metabolites. Another company in Boston is focused on Vionella, and they think Vionella is interesting because it uses up lactic acid, and lactic acid is associated obviously with muscle strain and the feeling of tiredness, though in this case the Vionella is present in the gut, so just exactly how that's contributing to athlete performance needs to be teased out a little bit. And then in other instances, you'll have some athletes that have lower levels of bifidobacterium and lactic acid bacteria. And I emphasize those because you might be more familiar with those names, because those are the sort of microbes that you typically find in probiotic products, bifidobacterium, and in particular the lactobacillus type of LAB. And so if you have an athlete that has low levels of those, there's a very easy potential of addressing that through getting them the right probiotic supplements. And there are very many other new and emerging species and strains that are of interest, but just to give you an initial introduction, I'll leave it there. So I think that concludes the first question, and I think we'll pause now because I think you might have some questions for me.
Thank you so much, Paul, for this excellent introduction to the field of the athlete microbiota. But I would like to know more about this topic. So what contributes the most to the athlete's enterotype? Is the diet, or it might be the type and the intensity of exercise?
So over the remainder of the talk, I hope to answer your question. So again, I'm going to give away the final plot, the final story, to say both. So both are important, and you can change the microbiota through exercise, and you can improve it through diet. But the perfect scenario is to have the best of both. The effect are at least additive and potentially synergistic. So like we, in the past, have worked with different groups of athletes that face the same challenges. So some athletes will have to make weight, or are involved in sports that require a lot of endurance. But sometimes, despite doing the same sort of training, depending on, for example, their socioeconomic background, they will eat quite differently. And so some of those athletes are very much inclined to take in information about their diet and will change their diet and eat what they're advised to eat. And then others, it's quite a job to try to convince them to not eat fast food constantly. And if they have a burger with a slice of lettuce, they'll remove the lettuce rather than having some fiber in their diet. And so in that case, they've done all of the right things with regards to exercise, but there's a big win for the group of athletes that don't have the right diet. Or in other cases, we've worked with teams where, as I've shown earlier, some athletes aren't consuming the right diet, but by educating them about that and having them change their ways, you can bring about a dramatic change. That's not to say there's other more subtle changes where you already have somebody who eats well and exercises well, but maybe they could be doing it slightly better, or the advice being given is good for a generic rugby player, but maybe they have some other underlying issue or some susceptibility or sensitivity, and in that case, maybe we need to get the right diet to them.
Thank you.
I had a question about training gut microbiota to make it resemble that of an elite athlete. Can that be done? And why is that better? Why should I do that? So
that's a great question. And so if you think about it this way, so if I do a fecal transplant, like we talked about for the animals, right, you can actually do that from humans for C. diff disease and so on. So somebody has as a last resort, if somebody has a very undesirable microbiome and they have recurring diarrhea, in some cases you take the fecal material from one individual and give it to another and make them healthy again by competing and removing that pathogenic microorganism. However, if you think about that in terms of an elite athlete, so they have a microbiome, or ideally have a microbiome that's perfect for their scenario, where they want to extract energy efficiently from food, keep them fit, have the right level of pro and inflammatory effects. So if we have a new up-and-coming athlete that ultimately wants to get on the team and perform like them, then yes, there probably is a certain merit in bringing their microbiota along and training it to get to that point. But if that transfer or that change or that modulation is taking place in somebody who's a couch potato and sits in front of the television all day, then that microbiome could be the worst microbiome for them, because they now have a microbiome that even more efficiently extracts energy from food. They're already probably eating more food than they should be because they're not exercising. And now they're even better at taking calories out, and so they're putting on more and more weight. Now, they may gain other side effects or other beneficial effects along the way in terms of immune stimulation or something else, but you do need to be careful in terms of how you train. Something else we've often talked about doing, but it would take a longer term arrangement with different teams, is to look to see what actually happens over a very long time, a very long longitudinal study, to see with, say, 14, 15-year-old players as they come in, is their microbiome already a little bit different and they have an advantage that helps them to excel later on in life? Or do they have the same microbiome as every other 14-year-old? But as a consequence of training and the dietary advice and whatnot, does that ultimately become elite and ultimately become perfect for their needs subsequently? But I think more research needs to be done in order to get to that point.
Thanks again, Paul, for your answers, and let's move now to the second part. So can you describe the interplay between the gut microbiota and exercise?
Yeah, so I'd be happy to do that. Okay, so I'm going to refer back to the Rugby study that I mentioned earlier. And this was the first time that we looked at this. So as microbiologists, we were faced with challenges. And I guess for people in the audience, this wouldn't be such a challenge. But for us, it was our first time doing anything like this. And our challenge was, for the control group, some of them were relatively healthy. I was one of the control group, and I run a lot, and I like to do marathons, so I was reasonably healthy. But lots of the others weren't and didn't exercise a lot. So we had to get a sense as to how much exercise those individuals undertake. And so the sort of standard questions involved: how much walking do you do, how much cycling, and so on and so forth. And we did this for the low BMI and the high BMI group. But there was no point in asking these questions of our elite rugby players. So how do we get a proper sense, or how do we monitor their level of extreme exercise, and I'll emphasize extreme, relative to those of the general public? So what we did is we used creatine kinase. And again, I'm sure lots of people could tell you about creatine kinase much better than me. But ultimately, it's creatine kinase, or CK, is an enzyme that you find in muscle. And when muscles are damaged due to extreme exercise and micro tears are introduced, the creatine kinase leaks out and enters into the plasma and can be detected. And in fact, it's a biomarker that's used for detecting a heart attack in individuals. And indeed, when we sent our samples from the rugby players who were going through pre-season training to our local hospital who did this testing for us, they rang us up very quickly and said, who are these guys? Do we need to send an ambulance somewhere, because their creatine kinase levels are crazy high? Because they had been really engaged in very intense exercise. And also you find very high levels of creatine kinase in marathon runners, people who are doing lots of downhill running, lots of weight training. And as you might expect, the levels were much higher in this group relative to the controls. So to see how and if exercise was correlated anyway with microbiota, we looked at all sorts of different metrics. We looked at lots of other things as well. But I'm emphasizing this because this is the one that hopped out and showed some correlation. These are all different metrics of alpha diversity. And in each case, we can see increased microbial diversity with higher levels of CK. In other words, more exercise correlated with increased alpha diversity. And as I mentioned earlier, alpha diversity is desirable. High alpha diversity is desirable. Now, coming back to some of the questions asked earlier in relation to getting the right microbiome in an athlete, and also how to train the microbiome, I'd also add another layer of complexity here, in that what's the right microbiome for a runner might not be the right microbiome for a rower, or for a boxer, or a soccer player. And this emerged from a study we did with a lot of Irish athletes, many of whom went to the Rio Olympics. So these were 35 different individuals. We didn't have a large enough N number to be able to do very good statistics on each sport in isolation. So instead, what we did is we broke down the sports into different groups. And this division is on the basis of increasing amount of endurance. So for example, boxing or rowing relative to gymnastics, or static component versus a very static scenario where you have golf or cricket, or much more mobility requiring scenarios like gymnastics and weightlifting. So when you look at those different categories, you can begin to see different patterns emerging. So these are the different sports that fell into the different categories, how many individuals we had in each instance. So if you look here in terms of C3, and to bring you back over, C3 refers to athletes from triathlon, rowing, boxing, cycling. We had high levels of another microbe that I'm introducing for the first time, a bacteria called Bacteroides. In C1, so C1 is this group down here, race walking, marathon, field hockey. We had higher levels of Faecalibacterium, and this was one of the ones I mentioned earlier as being a producer of short-chain fatty acids. Lactobacillus and Bifidobacterium are frequently found in probiotics. And then in this other B category, which is encompasses a broad variety of different types of sport, you have other different microorganisms dominating as well. You can also start looking at beta diversity. So, and as I mentioned earlier, beta diversity doesn't just relate to the names of the microorganisms present, but it can also relate to their functional potential, so what these microorganisms are capable of doing. And we can also look at metabolites. So when the microbes have different genes that produce different things, that will become apparent in a person's fecal samples. So you'll have higher levels of short-chain fatty acids or other different metabolites, and this can be determined by carrying out an NMR analysis of feces. And we also did a metabolomic analysis of urine, which is generally more reflective of differences in the host, or genetic differences in the host, rather than differences in the microbiome. And so from that, we saw, as it depicted here, lots of different separations, different groupings based on the different types of sports. Now, this was a cross-sectional study, and as I mentioned earlier, there might be instances where doing a longitudinal study might be more interesting. So just as a proof of concept exercise, we did this, but just with 2 individuals to prove that it was worthwhile. So I talked about couch potatoes earlier. In this case, we actually recruited 2 couch potatoes, 2 people that didn't exercise hardly at all. And they don't mind me using this term. I got permission to call them that. They were quite kind in that regard. So neither of them did very much exercise before we recruited them. We convinced one of them to take on a marathon. So that was quite an undertaking for somebody that didn't do much or any running at all. And the other person agreed to do a triathlon. They initially did a sprint triathlon and eventually up to an Olympic distance triathlon. But rather than just taking a sample at the beginning and another sample at the end, they were very kind and gave us fecal samples at multiple different locations along the way, multiple different time points. And so we were able to get a sample from this individual before and after he went skiing, and then he did a mountain race, and then he did a very long run over 20 miles, and then he reduced his training and he went kayaking and he got sick, and this other person took some anti-inflammatories and he had a head cold and so on and so forth, and we got to see what real life did, or what were the various different impacts of all of that on their microbiome. Now, as you might expect, because they didn't exercise much already, they did have an improved body composition, and they did have improved cardiorespiratory fitness. So what happened to their microbiomes and their alpha diversity? So you can see a gain or a loss in diversity at different times. And then you can also see how that changes in terms of the different pathways that are encoded as well. So the microbes and the pathways, but the 2 of them more or less reflect one another. So this person's diversity was down during the week of skiing. Maybe they had a bit too much to drink during that week of skiing and had too much of a good time. Their microbiome went up around the time of the marathon, but then lots of people do get sick after a marathon. He became ill. His microbiome dropped down to baseline levels as well. He had diarrhea. His microbiome diversity dropped even further. This other individual was up and down. He took an anti-inflammatory after a head cold. The head cold was associated with a reduced microbiome diversity. He took an anti-inflammatory and it increased. This would be something to maybe tease apart a little bit more with a larger cohort to see if that's a consistent finding, and ultimately completed the triathlon. this is really anecdotal. You can't extract too much information from that you can translate in terms of telling a particular athlete they should do this or that. But I think this is the way forward in terms of elite athletes, where you don't just do a microbiome test and that's the end of the analysis. Unlike a genomic test, where that is relevant for the rest of their lives, a microbiome profile will change, sometimes abruptly as a consequence of something that happens in life, and sometimes you want it to change for the better, so you want to see if your intervention has brought about an improvement in somebody's microbiome. So in this regard, you need to be able to monitor a microbiome change over an extended period. And so I hope that that's given you some level of insight into the impacts of exercise on the gut microbiota.
So you say these numbers are staggering, 100 trillion bacteria. I'm carrying 2 kilos of the stuff.
Yeah.
It's responsible for 90%, or can be related to 90% of the illnesses we get. And the exercise and intensity of exercise can modify this gut invariably to lead it to a beneficial effect. Can you comment on that?
So I wouldn't say invariably. So again, with any intervention, there are extremes. And so we have done some studies that involved especially endurance athletes who are probably overdoing it. It's more apparent in female athletes who have this REDS phenomenon, relative energy deficiency syndrome, where they're not getting enough energy. They're so conscious of their diet that they're not consuming enough calories, and that impacts on their immune health and whatnot. But also we see a microbiome component there. So the more and more exercise they're getting doesn't result in diversity to increase forever. Ultimately, there's a tipping point, and after you go beyond that, you're going down the other end. And so you're having a detrimental impact. And then there's other subtleties. So, and I guess I alluded to that in terms of the different athlete groups. So I think the studies still need to be done in terms of what sort of exercise will be best for particular individuals and particular sports in terms of having a beneficial impact on their microbial profile and diversity. In that, so is it weight-bearing exercise, resistance, or endurance, or a combination of both, or does it vary depending on the sport? And I guess ultimately more and more labs have started to get interested in this area, but there still is an awful lot of unanswered questions, and we alone haven't been able to do enough studies to do as much as we like. But I think those sort of questions bring us to the next frontier, and I think will help to make the breakthrough and to making all of this much more translatable in the future.
Okay. Thanks for highlighting these interesting aspects. So based on the interplay that you have just discussed, which one might be the most promising bacteria taxa to be monitoring when analyzing the gut microbiota in an athlete?
So in that regard, and maybe it's not avoiding the question, but it might seem like it, but ultimately I don't think it's as simple as that, unfortunately. There is no one keystone microbe. Some of the ones I listed are important. So high levels of Akkermansia, we're consistently seeing that across multiple studies. It's too frequent to be coincidental. And people have studied the various different components within the Akkermansia cell surface and the different metabolites it produces to try to figure out how that might be beneficial. So there's a lot of evidence accumulating there. But similarly, there's more evidence relating to Faecalibacterium. There's other microbes associated with a lean phenotype called Christinellacea, which we won't even get into because most people can't pronounce it. But so there's more and more emerging. But I guess the challenge quite often is in terms of the athlete microbiome, and indeed gut microbiomes in general, it's often quite clearly evident where it's suboptimal, when there's something wrong. But there is a broad range in the middle, away from the extremes, whereby you can have two individuals with quite a different microbiome, but they are equally healthy. And in some regards, maybe it comes back to what the microbes are capable of doing and their functional potential rather than the names of the microbes. Because as long as you have those key pathways to produce a short-chain fatty acid or to educate the immune system in some way, that which microbe is doing it might not be so important, but once that key ability is there, then that's the key thing. Despite that, I don't doubt that there will be increasingly companies harnessing this information to make the next generation of probiotics and biotherapeutic agents to cater for this market and to, in certain scenarios where there's a microbe that's regarded as desirable and it's depleted in an individual, that they would like to give that to the individual. I think the tests will then need to be done to see whether that improves their performance. Right now, I think the intervention involves that I would advise quite frequently would be a dietary one. So rather than changing one individual microorganism, change all of the microbiota for the better. Take a fermented food, take fiber, take the right type of fermented food for your particular needs, and that's where you can have a great win.
I think this is leading up to my next question, is about, we take for granted people who work here with our athletes are doing all the exercise they can do, which is reasonable. What can we do to make their optimum gut microbiota through dietary infection? What can we do for them?
Quite a lot, I would say. So I don't doubt your athletes are already consuming fantastic diets which are designed to get them the right amount of protein, the right levels of carbohydrates, healthy fats, and so on and so forth, all of which are important. But perhaps there's a few that need a bit of encouragement. And we've taken advantage of that among the rugby players, for example, whereby we've looked at the microbiome, we looked at diet, and showed instances where poor diet or not quite optimal diet has been associated with a poor microbial diversity. And these individuals, as you know, are so competitive and so looking for that 1% advantage that we can take that information and help them to help themselves, to encourage them to consume the good diets you're already encouraging them to eat. However, there are particular dietary components that are specifically catered towards having a good gut microbiota that the individual athletes might already be consuming, but maybe could consume an awful lot more. So if you think about fiber, the fiber we consume nowadays in the recommended daily allowances are if you consume that, it's good for your health. But if you compare that with, for example, Hazda warriors and tribes living in rainforests and whatnot, those individuals have a microbial diversity that's just off the charts, and they're eating all sorts of different foods, have much higher levels of fiber. One of my colleagues back in Cork, Jens Walter, is very much focused on fiber, and so he's doing some trials now where he's giving individuals extremely high levels of fiber, but the sorts of type of fiber or levels that we would have consumed hundreds of years ago, thousands of years ago. But as we've evolved, we now eat different diets and have more convenient diets. So, and he's getting results that are showing that returning back to those high fiber diets are having a beneficial impact. As I said, I'm biased. I'm very much a fan of fermented foods, but just eating tons of fermented foods might not cater for your need. For example, in our lab, we've shown that there are all sorts of different types of, one example of a fermented food, milk kefir. It's a fermented milk, and lots of people that make it at home. You have a milk kefir grain, which contains all sorts of different bacteria. You pop that into your milk, you ferment your milk, you drink it, the milk kefir grain is left over, you pop that into fresh milk, and you drink this kefir. Yeah, different ways of pronouncing it, but kefir. So that's passed down through different generations. I have kefir in my kitchen, you might have kefir in your kitchen. Lots of people are consuming it, getting in lots of good microbes, and that's good in general. But we've established that the microbial community in my kefir at home, it actually has the right microorganisms to reduce cholesterol and weight. We've also established that there are other kefirs that are very good at addressing anxiety. So I probably need more of the anti-anxiety one that my partner has or my friend down the road has than the anti-obesity one, because I exercise a lot anyway. But in that case, you just need a lot of good luck to get the right fermented food. But I envisage a situation in years to come, and we're already moving towards that, where for the kefir that is good at reducing weight gain and cholesterol, we've been able to pluck out the individual microorganisms present therein, put that back together to produce a new product in a manner that you're not just hoping you make the right one in your kitchen, but you can make it on a large scale, but without the standard sort of consequences of large-scale production, which is typically not having a lot of microbes in there or having a very boring product with not much flavor. That if you can take the real benefits of a true artisanal fermented food and recreate it in a manner that's scaled up, then you can have a food that's much more flavorsome, but you're also taking the best health benefits from that food and getting it to the right individual. And a different food, a water kefir or a cheese or a kombucha or a sauerkraut, might be better for somebody else.
So, for example, kefir for everybody. Okay.
Kefir for all.
If we had
I'll
kefir for all.
explain what I mean by kefir for all.
And then probiotic supplementation, same idea.
Yeah. So I wanted to expand on kefir for all briefly because you gave me an in to mention
Okay.
it. So kefir for all is a project we've done back in Ireland, which is one where we've taken some milk kefir, the same large batch of milk kefir, and we've disseminated it out into the world. So we've had lots of secondary school students ferment their own kefir at home and treat it almost like a Tamagotchi. I don't know if, maybe not many of you are old enough to remember Tamagotchis, but they had to mine their fermented food and subculture it and keep it healthy over 6 months and give us back samples every month so we could see how the microbial community in the kefir evolved and changed. But that's a story for another day. But coming back to probiotics, so much exactly like the example with fermented foods, it's no harm to take a probiotic. There's generally not any detrimental impact, but taking just a random probiotic might not do much for you. If you're already quite healthy, it might not do much. If you have a deficiency, it might do something, but what it does depends on the particular strain that's present, or strains that are present in the community. So there are strains out there that are very good in regards to controlling irritable bowel syndrome, and it's great if you have IBS to take that probiotic. But quite often, a lot of medics, when somebody has IBS, they recommend, oh yes, take a probiotic. And so the consumer knows no better. They go to the nearest supermarket, take a probiotic off the shelf, and wonder why it doesn't improve their IBS. Or worse still, the medic doesn't know which probiotic to recommend, and that's understandable because there's so much information out there and so much noise and so much marketing that the correct probiotic is probably out there, but there needs to be an education piece where that information is getting to the medic to tell them this is the probiotic to take or to recommend to that particular individual.
So it's how we can optimize the gut microbiota through dietary interventions.
Okay. All right, so we'll wrap up with this. And so some of this is kind of a logical conclusion from what I've presented already. So I mentioned earlier our elite athletes from the rugby study 10 years ago. We did a food frequency questionnaire with them. And so we were able to see what they were consuming. So we can put that in terms of macronutrients, how much energy they're taking in, how much protein, how much fat, and so on and so forth. However, the key consideration here that I draw your attention to is the percentage of total energy they get from the various different macronutrients, and most importantly, the fact that the elite athletes were taking a greater percentage energy from protein than people in the general public. And that's not so amazing because they're consuming more chicken, they're taking whey supplement, and so on and so forth in order to help their muscles to develop, especially among rugby players. Again, though, because we had all of this information and other dietary information, we were able to pull all of that together, go back to our microbiome data, and see what sort of things we could find. So again, I'm picking out alpha diversity, not just because I'm very interested in alpha diversity, but it was the one where we saw correlations. So alpha diversity can be measured in different ways, as I mentioned earlier. These are 5 different metrics of alpha diversity, and in each case, we're able to see some level of correlation between increased alpha diversity and more protein consumption, which leads us to think that protein is one way in which you could potentially, over time, increase your microbial diversity, and that's something we need to tease out a little bit more as well. Now, we also did other analysis which gives us an insight into various different impacts, and I refer back to some of our shotgun sequencing data. So what we did in this instance is to look not just on the names of the bacteria, but the different pathways that are encoded therein. And so again, we saw higher diversity in alpha diversity in terms of metabolic pathways in the athletes. We saw also differences in beta diversity in this regard. We looked at metabolic profiles. So again, fecal samples that were representative of the gut microbiota and its metabolites, and urinary metabolites that are representative of the host. And then some collaborators at Imperial College London did this analysis for us. And they rang us up immediately. Normally they'd send us an email and send us on the results and whatnot. But they were jumping up and down and really wanted to know more about what we had done to these individuals, because they said before they had studied these samples, they had never seen such a clear distinction between any two groups in society, with the exception of people who are really very, very ill and healthy. So these were healthy, but were elite athletes versus controls, and reflective of changes in their metabolites and the microbes in the gut producing those metabolites. So what are those metabolites? And there are too many to talk about, just like there are too many different individual microbes to talk about. But I did promise you earlier I would return to short-chain fatty acids and tell you why those are important. So short-chain fatty acids, SCFAs, are produced by specific microorganisms. They can help to alter chemotaxis and phagocytosis, so improve your immune cells to carry out those actions. They can induce reactive oxygen species, change cell proliferation and function in the gut epithelium, have anti-inflammatory effects, anti-tumor effects, antimicrobial effects against bad bugs. They can alter the gut integrity, so give you better barrier function, less gut permeability. And indeed, they can even be used after they've been produced by the microbes in the gut as an energy source by the epithelial cells in your gut. And now there are different types of short-chain fatty acids, so acetate, propionate, butyrate, isobutyrate, and so on and so forth. And we were able to look at those and compare them with dietary intakes and microbial diversity to see what correlated with what. And ultimately, it was revealed that some short-chain fatty acids were dictated by what you consume in your diet. These were the acetate, propionate, and butyrate. And others were correlated with bacterial diversity, so isobutyrate, isovalerate, and valeric acid. So armed with all of this vast amount of information, and again, coming back to one of the questions I was asked earlier as to whether it's diet or exercise is more important, we felt that we now were in a position to analyze all of our vast amount of data to really tease all of that out. So ultimately, 3 different things could be happening, 3 different models that we proposed. And so we began to try to tease those apart. So model 1 was being tested by assessing the impact of diet. And we could do that because we had control groups with a poor diet, and we had control groups with a high diet, and we were able to see what happens to their microbiome as a consequence. Now, we could also assess the effect of exercise because we had a control group with good diets and athlete groups with good diets. A small few with not so good diets, but generally good diets. So these both had good diets, but differed significantly in terms of their exercise levels. And then a third scenario is where exercise and diet both have a very important impact, and that's model 3. And how we could tease apart model 3 is to compare the extremes in both regards. So high levels of good diet, high levels of exercise, so this subgroup of athletes, and this subgroup of controls who had a poor diet and also didn't exercise very much. And so I already gave away the conclusion earlier by showing the combination of both. Model 3 is most important. In other words, diet and exercise in combination most dramatically impact the fecal microbiome as well as microbial and host metabolites. So you're probably going blind trying to figure out what's happening on this slide. So I'm going to explain it more slowly for you. So what you're looking at here is model 1 across here, model 2, and model 3. And we're looking at urinary metabolites, fecal metabolites, short-chain fatty acids, and alpha diversity of the gut microbiome. So first of all, I'll bring you back to the urinary metabolites. So you're seeing a differentiation clearly between those 2 groups, much more so than the other 2 groups when it comes to the combined impact of looking at diet and exercise. So model 2 was looking at the exercise effect alone, model 1, diet alone. Okay, so what happens with our fecal metabolites? In this case, you do see some levels of separation in all cases, but it's most apparent in models 2 and 3, so it's suggesting in this case that maybe exercise has a slightly more profound impact on diet. But when it comes to short-chain fatty acids, these important metabolites that are produced by gut microbes in the gut, you're seeing in both cases here, model 3 is the most relevant one. In one instance, exercise is important. And then finally, in terms of alpha diversity, as represented here by Shannon diversity, it was only the combined effect brought about a significant change. So thereby highlighting that the combined role of diet and exercise surpasses the individual effects of both in isolation. So really I'm getting to the end of the presentation now, but I didn't want to leave without giving you some final take-home points. So to re-emphasize a couple of things, I hope I've demonstrated to you that the gut microbiota contributes significantly to health and disease, not just in athletes, but across society in general. And that diet, and now finally exercise, appear to be modulators of the gut microbiota, but that the impact is most pronounced and most apparent when they're combined. That there's great potential to use diet and exercise to improve the microbiota in non-athletes and also in athletes. And there's merits of ongoing microbial analysis in athletes to look at various different cross-sections of communities, different types of athletes, because our studies were important, but more of those need to be done. But I really think that more longitudinal studies, large group of athletes, but over time to see how they change as they get older, as they go through different training regimes, what happens during pre-season rather than the rest of the year, what happens if somebody takes an antibiotic, how quickly do they bounce back from that? Do they bounce back naturally and we don't need to do anything to modulate their microbiome? Or has that antibiotic had a detrimental effect and we now need to help them along? I didn't mention what happens if somebody takes 2 antibiotics. They take one, they recover, they have to take another one a month later because the infection comes back. 2 doses of an antibiotic is really detrimental, and you have instances where people never return to their original microbiome after that sort of a stressor. So all of those sort of individual subplots are important considerations as part of the much larger story. There's lots of people that I need to acknowledge, but most notably Dr. Orla O'Sullivan, so my colleague back in Teagasc with whom all of this research was done. Lots of fantastic PhD students and postdocs at Teagasc and also at University College Cork. Collaborators in the Irish Sports Institute, Sharon Madigan at Imperial College London. My colleagues in the spin-out company, Microbiome. Lots of different sporting organizations, lots of athletes, elite and non-elite, and indeed lots of control groups, all of the various different institutes. And finally, thanks so much to all of you for your attention and for the very kind invitation. Thank you.
So thanks again, Paul. And before Dr. Mihic provides some concluding remarks and close the event, one last question. Okay. Which nutrients or dietary compounds can promote a healthier gut microbiota, or which one can disrupt it more easily? Okay, so I'm going to preach once again about the benefits of fiber and of fermented foods. I think both of those can have very profound benefits. So fiber, especially resistant fibers, so what you're considering there are these are nutrient sources that our body can't use. We can't degrade them and into smaller compounds and extract the nutrients, but the microbes, so that makes them bioavailable for the desirable microorganisms in the gut to take advantage of them and to encourage the growth of the right microbes and increase diversity. Fermented foods, so there are microbes in the foods themselves that will have a benefit, but they also produce lots of different metabolites. Very many of those foods also have other components within them. The microbes during the fermentation process release other nutrients that would not be present in the original substrate, so in the cabbage that's converted into sauerkraut and so on. So those are all desirable. I would also say there's lots of emerging evidence regarding the benefits of a Mediterranean type diet over other diets. In terms of negative impact, then the kind of more Western or industrial diets with high fat, high levels of salt, high levels of calories are all regarded as being undesirable. And you have scenarios where people move from parts of the world to Western cultures
and they're coming from a scenario where they have a good diet, they go to somewhere else, they can't continue to consume those diets, or it's more difficult to continue to do so. Their diet changes, and you see within a couple of months afterwards, their microbiome changes and their metabolic profile is impacted subsequently as a result. So there's lots of evidence of that.
Thank you.
So summary is a Mediterranean diet, kefir, fibers, probiotics. Any questions from the audience? Go ahead.
I would like to ask what is the positive importance of polyphenols for our microbiome?
Yeah. So that's a really good question, and I didn't mention them, and that was a mistake of mine. So I think there will ultimately be lots of evidence to support their benefits, but there probably haven't been enough studies yet. There are some people in my department, indeed, who study polyphenols and different polyphenol extracts from different vegetables and fruit and whatnot. And I've been trying to convince them to do some studies with us, initially just in terms of the desk-based exercises, but ultimately through some human studies. The little bit of evidence I've seen are positive, but there needs to be larger scale investigations. But I don't think there's any evidence of detrimental impacts, so I think any impacts will be positive.
Sorry, I got another question. So what is the importance of fasting?
Yeah, so that's an interesting one. That hasn't been looked at in terms of athletes. At least I haven't seen such papers. There's a bit of research starting to take place among the general public now, but not enough to my mind to have a clear answer to that question. So I think it's an important area, but there's an awful lot more needs to be done before we have a conclusion as to the relative benefits or negative impacts.
Thank you very much.
Thank you.
Anything else?
Thank you very much for your presentation, really interesting.
Thank
My
you.
question is more regarding the body composition and its relation with this gut microbiome. Have you evaluated something regarding this point?
Yeah.
Muscle mass, function, or amount of muscle mass, amount of fat mass, etc.?
Yeah. So that's a really good question. So we did do DEXA scans on the rugby players, and that was important because we didn't want to rely on BMI, which is not a good marker of adiposity. We did do other things like waist-hip ratios and whatnot, but it was important to do the DEXA scans as well. So in that regard, I guess it wasn't a very heterogeneous group among the elite athletes because they all had quite a lot of lean muscle mass, even though the forwards are quite big guys. They don't have that much adipose tissue. They're big guys, but most of it is muscle. So we weren't able to discern any particular phenomena there, but it would have been interesting to do for, say, the Rio Olympians who are involved in the different sports to tease that out a little bit, but we didn't have the opportunity to do DEXA scans there. But it's an intriguing question, one I'd like to return to in future studies. Thank you. Yeah, yeah.
One question I have, a little bit off the topic, but you say that, for example, 2 antibiotics at the same time is death, doom, and construction, but when I treat patients with H. pylori, it's inevitable. You'll get 2 of them.
Yeah.
What's your thinking of that?
So I guess when I said 2 antibiotics, what I meant is a course of antibiotics, and then a recovery period, and somebody gets ill again, and another course several months later. And so the impacts there are detrimental. With H. pylori, I would say in that case, because it is multiple broad-spectrum antibiotics or quite powerful antibiotics, you do need to take great care in terms of putting steps in place to help that patient or that person to recover and give them the right dietary interventions, probiotics and whatnot.
After therapy.
After therapy. Yeah, and even during therapy.
Okay.
Yeah.
Anything else, anybody? Before I go through, thank you very, very much, Paul Cotter, for coming all the way from Ireland to see us and tell us about this. We all know through metagenomics, gut microbiota has a huge impact on athlete health, his performance. We know that athletes have characteristic gut microbiota, which gives them competitive advantage, and we do want to nurture that through some of the advice you gave us. Getting the right gut microbiota promotes health, immune system function, synthesis of nutrients, absorption of nutrients, prevents disease. So we really have to take but it's such a new topic, you know, there's more and more research involved. When you say antibiotics or probiotics, how many are there? Which ones do we use? It's like you have to come back next week and give us another course on that.
Yes. Okay.
Anything else? Well, then let me conclude and thank you very much. This collaboration between Abbott and Real Madrid, I think it's good for us short term and long term so we can keep winning and bringing champions home.
Super.
Thank you, everybody.